Right, this is going to be a key to parts of the review. So for number one, really you can get those directly out of the notes, but some of the reasons cells might need to divide include if there is an injury that needs to be healed with new cells, replacing cells that are lost, like how your skin is always shedding cells and new cells coming up. For growth, so like from a baby growing into an adult, you need a lot more cells as you grow. And also for single-celled organisms for asexual reproduction. The cell cycle circle thing, we represent the whole time of a cell in one cycle in this circle. We have a small piece of the pie that is M phase. We have a larger piece that is G1, the first growth phase. We have a piece that is also smaller that is S phase. And then we have another little bit larger piece that is G2 phase, which is the second growing phase. And then altogether, G1 plus S plus G2 is interphase. Why might a cell stop dividing? Well, sometimes you just don't need more of that kind of cell. For example, in a fully grown adult human, you don't really need more bone cells. So the cells in your bones are going to stop dividing because unless you break your bone, you don't need more of those. Same with neurons, those just never divide again, most of your organ cells. So a lot of cells aren't dividing actively unless some kind of injury occurs, and some of them never can divide actively, and we call that non-dividing phase G0. The process of mitosis, I would recommend looking up some of the pictures of this, but basically the process is first the chromosomes condense up so they become visible and the nucleus goes away. So there's the nucleus going away. Here's my chromosomes condensing up. Then the chromosomes line up in the middle of the cell, and they are connected to these spindle fibers. Then the sister chromatids get pulled in half, separated to opposite sides of the cell, and then finally the cell divides with a new nucleus starting to form. That's the general flow of mitosis. And then how do cancer cells behave differently from normal cells? The main thing is they divide out of control. They divide and divide and divide, even when they're not supposed to divide. And that causes problems. That causes tumors. They could interfere with other cells that are trying to do their cell jobs. They can kill other cells. So yeah, generally, the problem with cancer cells is they just keep dividing out of control because of these mutations that they have. Right, so for all of these terms, these are definitions, and I'm going to let you find those in your notes. A lot of them are going to be in that cancer case study. But let's do comparing mitosis with meiosis. So first, and sorry I'm writing with my finger on this, but divisions, how many times do they divide? Let's do mitosis on the left and meiosis on the right. So in mitosis, cells divide once. In meiosis, cells divide twice. How many cells do you end up with? In mitosis, you end up with 2. In meiosis, you end up with 4. Diploid or haploid. Mitosis cells are diploid, meaning they have 2 copies of every chromosome, which is a full set of chromosome. Meiosis cells are haploid, meaning they only have 1 copy of each chromosome, so they have half the genetic material. We can also consider the type of cell that does this. So with mitosis, they are the somatic or just the normal body cells. With meiosis, they are the gametes, so producing the eggs and the sperm cells. Are the resulting cells the same or different? Are they identical or are they different from each other? With mitosis, the resulting cells are the same. With meiosis, the resulting cells are all different and unique from each other. With meiosis, we also have that crossing over that happens where the chromosomes will form a pair with their homologous chromosome and they will exchange bits with each other, so it's further just kind of mixing up the genes. Okay, so those are some of the ways that mitosis and meiosis are going to be different. Okay, I'm also not going to do all of the definitions. You can find those in your notes. Difference between codominant and incomplete dominant. Remember, codominant is both traits. Incomplete dominant is a blend of traits. And dominant recessive is the dominant trait covers up or hides the recessive trait. Let's do this cross for freckles. It says we have 2 heterozygous people with freckles, and freckles is dominant. Heterozygous means they have 2 different alleles, so they are big F, little f. Pull out this square. And it looks like we end up with phenotype-wise, the physical characteristics, 75% have freckles, 25% don't. Genotype-wise, we have 25% big F, 50% big F, 25% little f, little f. Next, we have a sex-linked trait, which is hemophilia. That means that it is carried on the X chromosome and not the Y chromosome. So we have a man with hemophilia. So a man is going to be XY. And if he has hemophilia, we can do little h on his X chromosome, no gene on the Y chromosome. And then we have a woman who is normal and not a carrier, so she will be X big H, X big H. When we fill out this, we should see that actually none of their children end up having hemophilia. We have their daughters will be carriers, but their sons will not have hemophilia. All right, this one is going to be one of the larger squares. We're looking for 2 traits at once. So we've got blood type and we have freckles. So you would need a 4x4 square. They are heterozygous for type A blood, so that means they are AO, because heterozygous means 2 different alleles, so they can't be AA, and O is recessive, and they do not have freckles. Freckles is dominant, so they must be little f, little f. And then their partner is homozygous for type B blood, so they are going to be BB, and heterozygous for freckles, so they are going to be big F, little f, and they would have freckles. So when I'm setting up my square, I'll put 1 parent on each side. For this first parent, half of their offspring are going to inherit the A allele and half are going to inherit the O allele, and then they are all going to inherit a little f allele because they only have little f alleles. For this parent, all of their offspring are going to inherit a B allele because they're homozygous for B, and half will get a big F allele, and half will get a little f allele. And then the question is asking what is the chance that their offspring has type B blood and no freckles. So you should do the entire square, but just to show you the answer to the question, these 4 squares are going to be BO, little f, little f. So these are going to be offspring that do not have freckles, which is 4 out of 16, which is the same as 1 out of 4, or a 25% chance that they would have type B blood and no freckles. A pedigree for a sex-linked recessive trait. So for pedigrees, I'm going to use circles for females and squares for males. And let's say this family has 3 children. Let's say they have a daughter and 2 sons, and then that daughter also has a son. And let's say that this mother here was a carrier, and then this daughter was a carrier. Maybe this son had hemophilia, and this son had hemophilia. So this pedigree is going to work for a sex-linked recessive trait. You can check and see if it matches up, but for example, this mother could be X little h, X big H, but she's a carrier. This father could be X big H, Y, and then this son inherited X little h, Y, so he has the trait. This son inherited X big H, Y. This daughter, X big H, X little h. This man would be X big H, Y, and then this son would be X little h, Y. And that's just one example of the way you could have structured a pedigree. You just want to make sure that if you draw like the Punnett square for any of these crosses in here, it matches up with what the results should be. All right, now let's look at this pedigree, and we're trying to figure out what kind of trait it is. So first thing I want to look at is, do I suspect that this trait is sex-linked or not? And just looking at it, I'm going to say I don't think so, because for one, it doesn't look like there's like more males that have it than females. And also we have male carriers, which we would not have male carriers with a sex-linked trait. So then we have to decide, well, is it dominant or recessive? And I see a case where, let's see, we have a father who is a carrier. They have children who are, they have a child who's carrier, who marries someone else who's a carrier. They have a son with the condition. So this is looking to me like a autosomal recessive trait. Because, well, first of all, we wouldn't even have carriers were it a dominant trait. And also we have a situation where we have 2 individuals who do not have the trait, but they have a child who does have the trait. So they must have inherited that recessively from them. How do we tell the difference between a chromosomal male and a chromosomal female on a karyotype? Well, the very last chromosomes that are going to be shown in the bottom right, those are going to be the sex chromosomes, and the X chromosome is much longer than the Y chromosome. So if you have one long, one short, that would be a chromosomal male, and if you have 2 long, that would be a chromosomal female. This karyotype right here, there is something abnormal on this karyotype. Normally you should have 2 of each type of chromosome, but if you look down here in the sex chromosomes, they actually have 3 sex chromosomes. They are XXY, which would be an abnormality. You could also have situations where maybe they have 3 of a different chromosome, which would be a trisomy, which would also be an abnormality, or maybe they only have 1. That would also be an abnormality in their karyotype. Drawing some DNA, I'm going to do it over on the side. So we have the pentagons, which represent the sugars. We have little circles, which represent phosphate groups. And those are making up the sides of the ladder. Then in the middle, we have our base pairs, which could be any of A, T, C, or G, but we have to remember that A and T must always go together. C and G must always go together. So if I was labeling the parts, I'd label the middle parts bases, or 2 of them together as a base pair. I would label the circles as phosphates and the pentagons as sugars. If I was going to circle 1 nucleotide, change colors, I circle 1 phosphate, 1 sugar, 1 base. So there is 1 nucleotide. Why is the order of base pairs in DNA important? Well, it's important because that's what determines like what the gene is, that's what determines what it does. The difference between you and someone else and the difference between you and another species is all down to the order of those bases, the order of those letters in your DNA. And the difference is where you might have an A but someone else has a C, for example. The double helix shape of DNA and the discovery, that's all in the Rosalind Franklin movie. But you want to know the names of Rosalind Franklin, Maurice Wilkins, Watson, and Crick, and sort of what they contributed to that, and also some of the, let's say, controversy surrounding what happened with the discovery of DNA and who got the credit for those things. All right, so that's all I'm going to go over in this video, but if you have additional questions, as always, please feel free to ask.